pathogens. And so you're likely to either be exterminated directly or outcompeted by sexuals, which are much better able to resist pathogens. And this is one of the best supported explanations, at least for the ecological, some of the ecological patterns that we see of sex and asexual in the wild. And in the lab too, there's evidence for this hypothesis. So Levi Moran and colleagues had this elegant experiment with appropriately enough C. elegans, where they co-evolved populations of nematodes with a virulent pathogen. And when they did this, they found that populations that were obligately selfing, so that were denied access to sex, were all driven extinct relatively quickly by this bacterium. And the interpretation was that when you're forced to co-evolve with virulent pathogens over extended time periods, if you lack sex, you're likely to go extinct. And so the question is whether this sort of short-term co-evolutionary lab result might reflect the longer-term fate of asexuals that we see reflected in these natural tippy-twiggy phylogenies. And the prediction of that hypothesis is that long-standing asexual lineages ought to be suffering major problems with pathogens. And so I've been trying to test that prediction with bdelloid rotifers, which are a group of microscopic, soft-bodied freshwater invertebrates less than half a millimeter long, not really visible without a microscope. And bdelloid rotifers are pretty diverse. There are more than 400 different species, multiple orders, multiple genera and families. And males are unknown for any of these species. The chromosomes appear to lack homologous pairs. There's low numbers of vertically transmitted retrotransposons, which we predict in selfish DNA theory when transposons can't move to new genetic backgrounds in an asexual system. And key sperm and meiosis genes apparently are absent. And so the inference from this evidence is that bdelloids are an exception to this rule that asexual groups are short-lived. Here we appear to have a whole class of metazoans that's been obliquely asexual for a long time. Just how long is unclear. We have a couple of sources of evidence. So there are some fossils in amber that have been interpreted as bdelloid rotifers from about 15 million years ago, but these fossils are really not very great. They're sort of like squashed Rorschach blobs where you think that's a rotifer, yes. So that's a little uncertain. Molecular clock estimates, we have those, but there are really broad confidence intervals because we're very unclear about the rates of molecular evolution in this group, and we have no calibration points because the fossils are, if they are rotifers, very ambiguous. So we're not really sure, but we can sort of say that it's probably tens of millions of years, even taking those estimates into account, so a long time. And that led John Maynard Smith famously to state in 1986 that the bdelloids remain something of an evolutionary scandal. How have they lasted so long without being wiped out by any of the mechanisms that we've come up with for the maintenance of sex? In particular, for today, how have they resisted the red queen? Shouldn't they have been overdue for extermination by pathogens? We can try and find some solutions to this apparent paradox. Maybe bdelloids are just really unusual in nature, they just don't have many pathogens. Hopefully that seems quite unlikely to you. And indeed, that's not the case. So bdelloids suffer from more than 60 described species of fungal and oomycete pathogens, predators, and parasites in a variety of different orders. So that's not the answer. Maybe these pathogens are not very virulent, so in many theoretical models of the red queen process, high virulence is required to drive the co-evolutionary cycling that maintains sex, and weakly virulent pathogens wouldn't be sufficient to drive an asexual lineage extinct. So maybe that's our way out. Here are some bdelloid rotifers that have been infected by some of these pathogens, and you can see they're all dead. These are just corpses with fungus sprouting out of them in all directions. And the reason they're dead is because the fungus requires the death of the host to spread its spores. So the virulence is always complete. It can't complete its life cycle without killing. This one is still alive, but this is where its ovary is supposed to be, and it's been replaced with just fungal spores. It's been castrated. So it's probably dead as far as Darwin is concerned. So no, the pathogens are all really, really virulent. How about this hypothesis? Maybe these fungi only attack the bdelloids opportunistically, so once in a while, or very recently. Maybe they've switched into bdelloid hosts in a very recent evolutionary time span. They haven't had long associations, long enough to drive this group extinct. Perhaps they're just occasional hosts. The prediction of that hypothesis would be that the pathogens should be phylogenetically scattered, showing a pattern of switches, multiple recent switches to bdelloids from their real hosts, which they've spent more time co-evolving with. So to test that, for the last 10 years or so, I've been gathering as many of these bdelloid pathogens from around the world as I can. We've got a pretty decent representation of the different species. Here we've got about half of all the described bdelloid pathogens You can culture them and extract DNA and build multi-locus phylogenies to test this hypothesis. Most of the interesting groups are in this fungal order Hypercreales, which you might know as the home of the zombie-making ant fungus Ophiocordyceps. Anyone who plays The Last of Us, the video game, will recognize that taxon. So we built phylogenies from this sample, and the main message is that multiple host shifts to bdelloid rotifers have occurred. So red here in this phylogeny is a bdelloid infected clade, and you can see there are at least 6 independent origins of bdelloid pathogenesis. 1, 2, 3, 4, 5, 6. So they have jumped from multiple different sorts of hosts in insects and so on into these rotifers. So that's consistent with our prediction, except for this one group here, which are all obligate pathogens of bdelloid rotifers. This group is called Rotiferoptera, rotifer destroyer, and it's diverse and monophyletic, which suggests there's been a long-standing association with the bdelloid rotifers. Just how long? Can we answer that question? Well, it turns out there's a really helpful fossil. The rotifer fossils might be squished and ambiguous, but there's a lovely preserved zombie insect in Cretaceous amber from 98 billion years ago. And we can use that to calibrate our phylogeny. And putting in the data, the crown age of Rotiferoptera is estimated at 46.2 million years, with pretty tight confidence intervals relative to the very wide ones we saw from the molecular clocks earlier. That means bdelloid rotifers as a group must be even older, because we can't have a pathogen radiating and diversifying before its obligate host. So that gives us a pretty solid estimate for how long these things have been around. And from the purpose of the red queen hypothesis, they've been associated with virulent pathogens for 47 million years, which seems somewhat problematic. New hypothesis. Maybe this is just an association and there isn't actual coevolution. So we might invoke some kind of ecological asymmetry where bdelloids don't encounter the pathogen often enough to be selected to develop resistance. It's a very one-sided sort of affair. That might explain how the association is possible for so long. So to test that, we've been gathering bdelloid clones of a couple of different genera, again from around the world, and we've been looking at the resistance and susceptibility to Rotiferoptera across these species. The way we do this is relatively simple. You just take 10 animals, stick them in a well, and dump on 1,000 pretty precisely measured spores of one of these Rotiferoptera pathogens. And you replicate this many times, kill tens of thousands of animals, and you measure the proportion at the end of the time that we become infected. And doing that, we can generate an infection matrix for species interactions here. And it looks like this. So you can see there's quite a bit of variation. This is the pathogen phylogeny along the top, this is the host phylogeny down the side. And there are some interactions that are very, very infective, almost all the rotifers die, and others where there's absolutely nothing at all. And there's quite a lot of patterning within this. Can we unpack this patterning a little bit? Can we use this data to test the hypothesis that the two parties have been co-evolving? Well, yes. I won't have time to tell you about the methods, but I refer you to this excellent paper by Jared Hadfield colleagues in 2014 about dividing variation in an infection matrix into the species, the phylogenetic contribution of the host, the pathogen, and whether or not there's a coevolutionary interaction terms significant there that would indicate that some species are very good at attacking particular hosts, and some hosts are particularly good at resisting some parasites and so on. And when you partition that variance, you find the strongest component is phylogenetic variance in pathogen virulence. So some clades of pathogens just are much better at infecting than others. But there's also a significant phylogenetic coevolutionary interaction, which means there has been some reciprocal selection on both parties in the system to resist the pathogen. And so we infer that bdelloids have actually been co-evolving with virulent pathogens for 50 million years without sex, at least on these macro-evolutionary timescales. And that seems problematic for the red queen. We might need to turn to some more exotic solutions to rescue our hypothesis. We can turn to theory. So there are several critical theoretical conditions for red queen cycling. Maybe, models are not entirely clear, but maybe this system lacks something like matching alleles or the negative frequency dependence that's needed to drive the coevolutionary cycles. We can turn to ecology. So in some previous work you can read about, we suggested that the dispersal ecology of bdelloid rotifers might help them to physically escape from co-evolving populations of pathogens in space and time. Or lastly, we might try to invoke genetics and suggest that there might be exotic genetic mechanisms that could replace the role of meiosis and help the rotifers shuffle their genes without needing sex. And that brings us to an interesting observation about bdelloid rotifers. So there's massive amounts of horizontal gene transfer from other kingdoms into these little animals. They've got, this is a well-established result, there are about 8% of their genes are not metazoan genes. They're genes from bacteria, plants, fungi, all over the place. However, even though this is massive relative to other animals, in absolute terms, we're talking about maybe 10 events per billion years. So really not that many, not nearly on the same timescale as sex. But it's tempting, given this result, to speculate that maybe these bdelloids are swapping DNA horizontally with each other, maybe at much higher rates, and maybe that's replaced the functions of sex and lets them do all the fun stuff that meiotic populations do. And recently, last year, there was a paper that claimed evidence for this hypothesis in Current Biology. Bortoli et al. Picked out some rotifers and sequenced multiple markers, and they found that some markers appear to belong to one species, and some of the markers appear to belong to a different species, and they inferred that there's been horizontal swapping of DNA between these species. They think there's strong evidence that bdelloids have replaced meiotic sex with horizontal exchange of DNA. And a comment on this, Tanya Schwander proposed that bdelloids should no longer even be considered as asexuals because of this horizontal exchange. Bringing you right back up to date, 5 days ago we published on bioRxiv a following preprint in which we demonstrate, at least in our view, that these results are actually explained by cross-contamination of the DNA of animals among the tubes. I don't want to talk too much about this because the author's not here to defend their work. But briefly, I suggest you go and look at the paper if you're interested. Briefly though, we looked at the sequencing chromatographs for some of their mitochondrial haplotypes, and we found that there were multiple peaks consistent with multiple mitochondrial haplotypes divergent by about 15%, which strongly suggests that there may have been multiple animals in these tubes. And with multiple animals, you can explain the horizontal nuclear gene transfer. So it's the same animal that apparently donated the genes of the nuclear loci, whose mitochondrial haplotypes turned up in the chromatograms. And also we point out that the distances over which these interspecific transfers are occurring by homologous recombination are about 75% identity, which is not credible for those molecular biologists and recombination biologists among us. It's too far, you can't get that kind of recombination. So in summary, bdelloids have been co-evolving with virulent fungal pathogens across macroevolutionary timescales. Seems problematic for the prediction of the Red Queen, but there are many possible solutions still unexplored. There's currently, in my view, no credible evidence that horizontal genetic exchanges between rotifers have replaced sex. That's still an open question. We need careful testing of that and other hypotheses. So in the words of Don Maynard Smith, the bdelloids remain something of an evolutionary scandal.